Good day to all of you teachers and classmates. A very warm welcome to all at our discussion about the plastids and vacuoles. For this discussion, we're going to talk about the plastids, types and function of plastids, structure of plastid, what is vacuoles, vacuoles in different types of cell, types and function of vacuoles, and structure of vacuoles. In the cells of plants and algae, there are double membrane organelles called plastids. These frequently contain photosynthetic pigment as well as pigment that may affect the color of the cell. The vast majority of plant cells are housed inside the vacuole, which is a membrane-bound sac. Other organism cells may include vacuoles, but they are much smaller and fewer in number. Learning objectives. At the end of the unit, I am able to define and identify plastids and vacuoles, discuss the structure of a plastid and vacuoles, state and explain the biological process of the plastids and vacuoles, and identify and describe the types of plastids and vacuoles. What is plastids? Green plant contain plastids. A single leaf of a plant contains numerous cells inside it, and one of them is plastids. Plastids are a class of large organelles that contain pigment or food and are found in the cytoplasm of cells. They have a double membrane and their own DNA and ribosomes similar to mitochondria in animal cells. They can be found all over in plants and other photosynthetic organisms. Organelles are specialized membrane-bound structures in cells that help their function and survival. They are to the cells like organs are to the human body. Plastids function in synthesizing and storing food for the plant to use. In addition, plastids allow the plant to make energy from sunlight, carbon dioxide, and water through photosynthesis. In order for plastid to accomplish all the food production and storage that the plant needs, there need to be multiple types of them. All plastids begin as progenitor for plastid that came from the meristem and later differentiate into the other types. Some develop into ethioplasts if the plants begin to develop in darkness. These are not full matured and cannot participate in photosynthesis, but they are distinct stage between proplastid and mature plastids. The four major types of plastids are chloroplasts, are green and serve as the sites for photosynthesis in the cells. Highly pigmented plastids called chromoplasts give plants the colors they use to attract pollinators. When the plant stops using photosynthesis in the leaves, the chloroplasts develop into gerontoplasts. Non-pigmented plastids used for storing starch, lipids, and proteins are called leucoplasts. Amyloplasts, these are double envelope organelles. For the plant, they store and synthesize starch through glucose polymerization. Proteinoplasts, they store protein and exist in seeds. Elaioplasts, they store fats and oils that they are needed for the plant, they exist in seeds. Structure of plastids. The most talked about types of plastid is the chloroplasts. These organelles are responsible for making plants green and producing energy for the cells and the plant as a whole. They are filled with stock of thylakoid discs called grana. Each thylakoid membrane contains the green pigment, chlorophyll, and the space within the disc is called the lumen. The fluid component that surrounds the grana and fills the chloroplast like the cytoplasm of a cell is called the stroma. Photosynthesis takes place in the thylakoid stalk as the energy captured from the sun is pumped across the membrane from the stroma to the lumen. The hydrogen protons rush back across the membrane into the stroma where ATP synthase converts that energy into ATP. The cell's energy currency, the ATP is built into glucose molecules to be stored for later use. What is vacuole? The definition of vacuole is a single membrane-bound organelle with no defined shape or size. It is one of the largest organelles in the cell. specifically in the plant cell. It is present in different types of cells like animal cells, plant cells, fungal cells, protein cells, and bacterial cells. The structure of vacuole is basically dependent on the individual cell requirements. A vacuole is a membrane-bound organelle that is present in all plant cells. Some animal and fungal cells also have vacuoles, but they are much smaller. Most mature plant cells have one large central vacuole that can occupy as much as 80% of the cell volume, making the vacuole the most prominent organelle in plant cells. By storing various materials, the central vacuole keeps its water potential as low as the cytoplasma potential and maintains pores against the cell wall. Turgor pressure is essential for supporting plants in an upright position. Plant and fungi both contain vacuoles. Among them, plant cells generally contain one large vacuole in each cell, called central vacuole. It is so enormous that it may consume up to 90% of cell space. On the other hand, fungal cells contain multiple small vacuoles. A thin membrane called tonoplast lines the vacuoles in both, helping it retain its structure and shape. Vacuoles in plants and fungal cells perform similar function in some ways. Though it mainly acts as a water reservoir, it also stores some small molecules such as polyphosphate, amino acids, several ions, and other small molecules. Also, as these cells do not retain lysosomes, vacuoles help them to break down materials. To maintain function is to maintain proper pH, stores water, maintain turgor pressure, and adjust size of the cell. In protists, they mainly use these vacuoles to regulate the water content in their cells. Freshwater protists tend to absorb too much water into their cells, which may lead to cell rupture. In that case, these contractile vacuoles prevent them from rupturing by expelling the excess water out of the cell by continuous contraction. In animal cells, as already mentioned, vacuole is not always present in animal cells. However, depending on the function and need of a cell, they may contain small vacuoles in each. Unlike plants, it is not surrounded by tonoplasts. Also contrary to plants, vacuoles in animal cells do not break down and degrade larger molecules as they possess lysosomes. Vacuoles in animal cells do not help in providing structure to the cell. Instead, they help in the transportation of several substances into the out of the cell. The vacuole usually provide two types of transport, exocytosis and endocytosis. In exocytosis, vacuoles accumulate waste products and actively transport them out of the cell. On the other hand, in endocytosis, cells actively transport several nutrients, toxins, cell debris into themselves with the help of vacuole. During the process, vacuoles are there, the substances are break down or stored while transporting. The main function used for storage and can contain nutrients, water, and waste. Soft vacuoles are the most common type of vacuole in plants and contain many substances like minerals, sugar, oxygen, carbon dioxide, soluble pigments, and organic acids. Cell sap has acidic pH and is hypertonic in nature and remain distended because of osmosis and make the cell turgid. The function, they maintain turgidity or turgor of the plant cells. Some acacia trees produce and store cyanide in their vacuoles and protect the cell from cyanide poisoning. They impart colors to the fruits, flowers, buds, and leaves due to the presence of anthocyanins and anthocyanins in the sap. Some sap vacuoles act as lysosome, having hydrolytic enzymes. Vacuoles of the stores cells and seeds stores proteins. The contractile vacuoles stop a cell from becoming too waterlogged and swollen to the point of rupture. We refer to this process as osmoregulation. Some excretory materials may be present in the water existing in contractile vacuole. As a result, it also serves as a means of elimination. The function of the contractile vacuole prevents too much water from accumulating in a cell and swelling into the bursting point. This process is called osmoregulation. The water leaving the contractile vacuole may contain some excretory products, hence it has an additional role of excretion. Food vacuoles help in internalization and digestion of food. The remaining undigested food is expelled out of the cell by the fusion of food vacuole with the plasma membrane. Gas vacuoles occur in some prokaryotes like bacteria. A gas vacuole is actually a group of microscopic vehicles, each having a protein membrane around it. The vehicles contain metabolic gases. The function of a gas vacuole is stores gases, regulate buoyancy of the cell, provide mechanical support, and protect the cell from harmful radiation. Structure of vacuoles. Cell wall. The cell wall is the outermost covering of plant cells. The function of cell walls protecting the cells against physical damage and invading pathogens, regulates and controls the direction of cell growth, providing the strength, structural support, and maintaining the shape of the cell. Cytoplasm removes waste materials, help in the cell respiration, and converts glucose into energy. The cell membrane is also known as the plasma membrane. The function of cell membrane is plays an important role in cell signaling and communication, protecting the integrity of the interior cell. Providing support and maintaining the shape of the cell. The size and shape of vacuoles vary according to the specific requirements of the cell. There can be one large vacuole in a cell or many small vacuoles. Plant cells usually have central vacuoles, whereas animal cells typically have more than one vacuole. The cell shaft is the fluid part of the vacuole, which contains enzymes, water, several salts, glucose, and waste materials. Tonoplasm is a phospholipid bilayer membrane that provides tensions and maintains the turgor pressure of the vacuole. It also controls the movement of ions in and out of the vacuole. Before we end our discussion, I hope you have all learned a lot of new things about the plastids and vacuoles. Thank you so much for being with me until the end of the discussion. We are really hoping you learn more. Have a great day. Keep safe, everyone. Always wear masks, maintain a distance, and avoid touching mouth, eyes, and nose with the dirty hands. Have a nice day and safe day. My name is Jonalyn Bernardo from BS Edu 3B, signing off.